So in this video, we're going to learn about nucleic acids, which are the molecules that store our genetic information. So as I said before, a nucleic acid is a macromolecule that stores genetic information, and nucleic acids are the only macromolecules that can reproduce. And because of this leads to the capability for the growth and reproduction of organisms. Because DNA can replicate itself, we can divide our cells and make new ones. If we didn't have that ability, we wouldn't be able to pass our genetic information, the instructions for building a new cell, onto the next cell, and then we would die. Once the cell died, there would be no more of us. So we need nucleic acids to be able to allow for us to continue growing and to continue passing our genetic information on to new members of our species. So nucleic acids are made of three components. One of them is a pentose sugar, and the pentose sugar can either be ribose, which is this one, or deoxyribose, which is this one. And the only difference between those two molecules is that on this carbon, so carbon number 2, there's a missing oxygen. Instead of having a hydroxyl group, it's just a hydrogen in this position, so we don't have the hydroxyl group showing. The second component for a nucleic acid would be a phosphate, and a phosphate joins to the pentose sugar. We'll talk about that a little bit later. The third component of nucleic acids is the nitrogenous base, and you're probably familiar with 4 of them already. So there are 5 actually. One of them is only found in RNA, and one of them is only found in DNA. So we have adenine and guanine are both called purines, so they have a double ring, and they're slightly different in their bonding. So like there's an NH2 on adenine and a double bonded O on guanine. And then there's 3 pyrimidines. So there's cytosine, thymine, and uracil. And thymine and uracil are extremely similar. The only difference is right here on this carbon, there's a methyl group or a CH3, and over here there's just a hydrogen. So thymine is found in DNA, and wherever thymine would be in DNA, it's substituted with uracil in RNA. We'll learn more about that when we study our molecular genetics unit. So all 3 of those components combine together to form the nucleotide, and rather than sketch out the individual carbons, hydrogens, oxygens, phosphorus, etc., we have a sort of schematic sketch that is universally accepted for the nucleotide. So we always represent a phosphate with a circle, the sugar with a pentagon because it's a pentose sugar, and then a nitrogen base for the adenine, guanine, etc. So the phosphate is added to the 5th carbon of the sugar, so up here it's sticking up in the air, and then the nitrogen base is added to the 1st carbon of the sugar, which is over here. And nucleotides just don't exist usually alone. They join together to form polymers. So they would connect at the phosphate of one nucleotide, so up here, and then the next one would combine down at carbon 3 of the next nucleotide. So it just keeps repeating over and over again. So right now we'll take a look at the structure of DNA. So DNA is a double-stranded molecule. So you can see here there's one strand going this way and another one going this way. And the 2 strands are held together by hydrogen bonding between the bases. So adenine and thymine can form 2 hydrogen bonds, and guanine and cytosine will form 3. And this, if you notice, one of them is pointing up and one of them is pointing down. So the 2 strands run what we say is anti-parallel to one another. So on the left-hand strand, the phosphates are pointing up. So we say that this is the 5' to 3' end because this is the 5th carbon, so the 5' carbon. And then at the bottom, they all align. This bottom part right here is the carbon 3, so the 3' end. So this strand runs 5' to 3', and the sugars and phosphates are pointing up. And then on this side, it's running from 3' to 5' because poking up here is the 3rd carbon in that sugar. So the 3' end is pointing up, and the phosphates and 5th carbon are pointing down. So this one goes 5' to 3', and this one goes 3' to 5'. Again, when we get to molecular genetics, we'll find out some details and some reasons why those directions are important. Now, Some other things about DNA. The sugar is deoxyribose, so that's where the D comes from. And we have our 4 bases are adenine, thymine, cytosine, and guanine. Now we're going to talk about RNA. So unlike DNA, RNA is a single-stranded molecule, so here we go. And in this case, the sugar is ribose instead of deoxyribose. And instead of having thymine, we have uracil. So it's adenine, cytosine, guanine, and uracil instead of thymine. So those are the differences between DNA and RNA.